Okay, so it is a pleasure for us to have today in our Bar-Ilan University Vision Science Seminar, Dr. Avital Hahamy from the Wellcome Center for Neuroimaging, for Human Neuroimaging at UCL. Avital did her BSc degree in biology and psychology with an emphasis on brain research in Tel Aviv University, graduating with honors. She then did a master's degree in neurobiology at the Weizmann Institute of Science under the supervision of Professor Rafi Malach, where she studied the relationship between functional connectivity patterns and schizophrenia. She then continued to do her PhD in Professor Malach's lab, where she continued studying clinical populations as autism and the Charles Bonnet syndrome using resting state activation patterns. Today, she's doing her postdoctoral research at UCL in Professor Tim Burns' lab. During her graduate and even her undergraduate studies, she already joined leading labs in neuroscience to work and collaborate with distinguished professors, including Professor Karl Zillis's lab in Germany, Professor Heidi Johansenberg's lab in Oxford, and Professor Marlene Berman's lab at CMU. Since then, she has collaborated with additional distinguished researchers, including Professor Tamar Makin from UCL, Professor Ro'i Solomon from Haifa University, and others. She won many prestigious grants and honors, including the EMBO Long-Term Fellowship, the Marie Curie Postdoc Fellowship, Human Frontier Science Program Fellowship, the Ben-Gurion University Award for Postdoctoral Women in Engineering and Natural Sciences, the Organization of Human Brain Mapping Merit Award, the Society for Neuroscience Trainee Professional Development Award, and many, many others. She publishes in top leading journals as Nature, Neuroscience, eLife, Current Biology, Brain, Journal of Neuroscience, and many others. Her work is often covered by the media, and she is invited to many invited talks across Europe. So I will stop here and invite you, Avital, to tell us about your exciting new research. And thank you so much for joining us today.
Thank you, Sharon, for the very, very kind introduction. And thank you for being here. I'm very excited to be telling you about my work today. And I'll be telling you about reactivation in the human brain that connects the past with the present. But actually, what I'm going to be talking about is how we make sense of our experiences. So, for example, how will you make sense of this talk? This is me giving a similar talk even before the paper was published in a lab meeting. And this is Alon. He's in my group, and he's listening to me and trying to understand what it is that I'm saying. And although this experience of me speaking is continuous, Alon's brain is actually chunking this experience into discrete events. This is according to the event segmentation theory. So let's assume that this is the sound wave of my talk. And just for the sake of argument, let's assume that these blobs of audio represent events, for example, slides. So this will be the first slide, the title of my talk. And then on the second slide, I'm going to tell you about the intuition to the big problem I'll be talking about. And when your brain notices this switch in context between the title and slide 2, it's going to mark an event boundary. That's the end of one event and the beginning of another. So at the end of slide 2, we're going to have an event boundary, and then maybe someone is going to make a joke. That's an entirely different context. So again, we have an event and an event boundary. And then on slide 3, I'm going to give you a literature review, event boundary. And on slide 4, I'm going to tell you what's still missing and how I propose to solve the problem. Now, at event boundaries, it is proposed that your brain represents the gist of each event, the bottom line of the just ended event, and it stores this gist into memory. And this is an offline function. We don't really need this to do anything at the present online. It's just to maintain a memory of that representation. But isolated event representations won't help you understand the narrative, because if we're in slide 4 and I'm telling you what's still missing, then you need to know what is known. And what is known was actually presented in slide 3. And if I'm telling you how I propose to solve the problem, you need to know what the problem is. And that was actually presented in slide 2. So I wanted to know, how do we bind these related events across time? And there are several clues that led my research. And the first is replay at event boundaries. So replay is a well-known phenomenon, heavily studied in rodents. And in the rodent hippocampus, also in our hippocampus, there are play cells. And these cells fire in specific locations in space. They have their tuning curve that is positioned in a certain location in space. For example, this red cell fires in this red location, and this pink cell fire at this location. So as the rodent progresses across a path, you would see different cells firing across different locations. But the interesting thing is that when the rodent pauses or receives a reward or at the end of a trial, and all these things are very similar to event boundaries, the end of one event, the beginning of another, a change in context, then the same cells fire again in a compressed manner, and the mouse is doing nothing. So this is what we call replay, this rapid reactivation of self-firing patterns in the brain of rodents. We also know it exists in humans. But interestingly, in humans, we know there are reactivations not only during spatial tasks, but also during movie watching. So in this study, this is an EEG paper, where they looked at the event boundaries as defined as cuts between scenes. And what they found was that at the ends of scenes, at the ends of events, they found a reactivation of the just ended event. And I'm calling this a reactivation rather than replay because in neuroimaging studies, we often don't have the spatial or temporal resolution to see this sequential reactivation of self-firing patterns. So we only call it reactivation. So back to our toy example. So at these event boundaries, where there is this cut between events in our experience, it seems that there is this replay or reactivation of the just ended event. And this reactivation is proposed to have an offline function, as I said before. It chunks events and stores them into memory. And to better understand what I mean by this replay, then I've just switched slide. That means that your brain should now detect an event boundary, a change in context. And what it should do is replay the just ended event, the previous slide. So it would replay the fact that there is replay in rodents during contextual shift and an ongoing experience. There's reactivation in humans during contextual shifts and an ongoing experience. And both phenomena are presumed to have an offline role. Okay, so first clue, we know there is replay or reactivation at event boundaries. Second clue. Both event segmentation and replay engage the default mode network. And this is a very well-known network. And for years, it's been termed the offline or task negative network because it tends to reduce its activity during active tasks and increase its activity during resting state. Although this finding has been refuted many times since, and there are many tasks that actually activate the default mode network. For example, event boundaries. So it turns out that when people either watch a movie or listen to a story, areas of the default mode network actually increase their activity precisely at event boundaries. And the three areas that are most replicable in doing that are the precuneus and retrospinal cortex, the angular gyrus, and the hippocampus. So there is increased activity at event boundaries in default mode areas, but interestingly, default mode areas are also highly activated and most activated in the brain during replay in rodents, in monkeys, and in humans. So there are convergent evidence that when there are sharper ripples, the times in which we find replay in the brain, or when we see replay, for example, according to this study, then the areas that are most active in the brain are areas like the retrospinal cortex and other nodes of the default mode network. So far I told you that there is replay or reactivation at event boundaries and that both event segmentations and replay engage the default mode network. Third clue, offline replay can bind related events. And this is a beautiful paper by Gupta et al. And they had rodents run in this very special maze. And in this maze, the rodents can either run through this compartment marked in arrows or through this compartment. But what the rodents never ever do is run through the outer loop. And then they have the rodent sleep. And during sleep, they see replay. They see replay these self-firing patterns that map to one compartment of the maze or to the other compartment of the maze. But the amazing thing is that they also see replay of this outer loop. the path that has never been taken by rodents. And they propose that what replay can actually do is piece together different experiences to create this model of space, of our environment. So if replay can take remote events and piece them together into a model of space, maybe it can do it for other types of experiences, like our daily experiences. In other words, could it be that the same neural mechanism that supports our construction of a spatial model would also construct our model of understanding our daily experiences? Now, this is a technical challenge. This is hard to prove because this would require us to actually detect this replay during naturalistic experiences. And hopefully throughout this talk, I'll be trying to convince you that I found a way to do that. So my question is this: if offline replay can bind related events, can online reactivation bind related events, like the ones in my talk when you're listening to my slides? So is temporally remote information reactivated at event boundaries of naturalistic experiences? So again, to explain what this means, here again, you have an event boundary. I switch slides. And now, according to my hypothesis, you won't only be reactivating the just ended event, the just ended slide. You'll be reactivating all the information that is needed for you to understand the hypothesis that I just presented. Is temporally remote information reactivated at event boundaries of naturalistic experience? Which information would that be? Well, the human brain parcels ongoing experience into events. That was slide 2. There's online reactivation of the current event at event boundaries. That was slide 4. But to make sense of ongoing events, temporally remote information must also be replayed. Slide 3. Rodent replay can link remote spatial events. Slide 7. Can human reactivation link remote episodic events? Cool. So experimental approach. We used narratives. And why did we do that? So I'm now looking for events, the brain representations of events, and I want to know how they integrate together. So if I were to use repeated events or experiments in which we have repeated trials, like rodents navigating through a known maze, or any experimental design that repeats the same events again and again and again, then the events are similar and their brain representations will be similar, and I won't be able to tease them apart. But in ongoing experiences like narratives, each event is unique and it would have a unique brain representation. So that's the huge advantage of using narratives like stories or like movies. So because this project was actually done during COVID, I used freely available data sets of people either listening to a story or watching a movie. And these two data sets are actually very, very different from each other in many aspects. The first is purely auditory. The second one is audiovisual. So the modality is different. The content is obviously different. The first is a drama, the second is a mystery. The narrative structure is very different. In the movie dataset, there is a linear structure. One scene leads to another in this organized way, whereas in the story dataset, we have two narratives marked in red and blue, and they're interchanging. And they're completely independent, different characters and locations and plots, and they're interchanging, and then they come together at the end. So it's a different narrative structure. The stimulus structure is different. In the story dataset that is well controlled, each scene or paragraph has a constant length. But in the movie dataset that is less controlled, we have very short scenes and very long scenes, and the pre-processing of these datasets are different. And the reason I'm boring you with all these technical details is just to say that given the differences between these datasets, any consistent effect across the datasets will tell us something general about the neural processing of naturalistic stimuli, because consistent effects couldn't be ascribed to a certain attribute of a certain dataset. Okay, so methodology, representational similarity analysis. I'm going to very briefly explain this. So the first thing I'm doing is detect event boundaries, let's say in a movie. Now, I can't use the actual frames from the movie, so these are my kids. So let's assume we have a movie scene with this going on and then this going on. And I'm detecting event boundaries as the cuts between scenes. And then because I'm using fMRI, I'm going to detect the TR, that's our measurement, our temporal measurement in fMRI, our smallest temporal measurement. I'm going to detect the TR that holds this event boundary. So an event boundary is a point in time. It's an instantaneous thing. And my TR is going to be 1.5 seconds. And it contains these instantaneous change between scenes. And now I'm going to take this TR, I'm going to shift it by 4 additional TRs, and that is 6 seconds. The reason I'm doing that is because in fMRI, we have this hemodynamical lag. So when you present a stimulus, it takes around 6 seconds or 40 hours in my case until you see this activation in the brain. So now I'm shifting my data, so each event boundary is now the peak of its hemodynamic delay. And now I'm going to do a searchlight analysis. This means I'm going to take each voxel, 3-dimensional pixel in the brain. So I'm taking each voxel, and for each voxel, I'm constructing a sphere around it. This is going to be the cluster I'm going to work with. And this cluster at a given time point, for example, at a certain event boundary, is going to have some level of activation. Each voxel here is going to have a specific level of activation. You can think about this as a mosaic. This mosaic changes across time. For different event boundaries, I'm going to have a different mosaic. So this is what we call, this mosaic is called a representation. Now, I have my timeline. EB now is event boundary. So for each event boundary, I have a representation. I'm going to represent it now as this vector with voxel that have different colors representing different levels of activity. And I'm also extracting the mean scene representation. So for each scene, I'm taking all the time points within the scene, all these representations, and I'm averaging them into a single representation. That would be my average scene representation. And this is actually very common in the naturalistic literature. Obviously, I'm going to lose all the fine-grained information that exists in the scene, but I'm going to be left with something more general, like the context or the gist, exactly the thing we think that the default mode network codes for. Okay, so this is about representations. Now let's talk about the similarity analysis. My hypothesis is that at event boundaries, you're going to link remote events. So basically, you'll be loading previous representations from memory to bind them with the current event. And that will basically mean that at this time point, at this event boundary, I should find correlation between this representation and representations of different scenes that came before, right? If they're uploaded from memory, then I should find a correlation between the current event, the current moment in time, and different events. So what I'm going to do, I'm going to correlate my event boundary representations with my scene representations, and that's going to give me a correlation matrix. Now let's talk about this matrix, for example, for a second. It has 2 parts. All these entries below the diagonal are the past part of the matrix. These are the correlations between each event boundaries on all the scenes that came before it. It also has a future part. These are the correlations between each event boundary and all scenes that come after it, okay? So lower triangular part is past, upper triangular part is future. It also has this main diagonal, which I don't care about. The main diagonal is the correlation between each event boundary and the just ended event. And we know there is replay there, so we don't care about that. We want to know whether I would find reactivation not between an event boundary and the just ended event, but with remote events. So for example, let's assume that this matrix represents my talk. In the beginning, I gave you an introduction to event boundaries, and then I told you there's replay at event boundaries, and then that there's replay of remote information. So when I'm telling you there's replay at event boundaries, to understand what I'm saying, you're going to need to reactivate the fact that there are event boundaries and what they are. So at this event boundary, you will need to reactivate this prior event. And when I'm telling you there's replay of remote information, you're going to need to know that there is replay at event boundaries, and you're going to need to know what event boundaries are. So these entries of the matrix should be reactivated at this event boundary. And indeed, if this is actually data from a single participant, you can see that indeed different entries of this matrix show different levels of reactivations. So some entries are more reactivated than others at certain event boundaries. But there's a problem here. because can these correlations simply reflect scene similarities? So I'm using fMRI, and fMRI has very low temporal resolution. This basically means that an event boundary that is very close in time to the scene that came just before it is going to be highly correlated with this scene. And this scene could be correlated with other scenes because they're similar. So if I'm telling you that I'm measuring the correlation between event boundaries and past scenes, in fact, what I could be just measuring is just scene correlations. If an event boundary is similar to a scene and a scene is similar to another scene, all I'm measuring is just scene by scene correlations. But if this is true, then this event boundary over scene matrix is not really an event boundary by scene matrix, it's a scene by scene correlation matrix. And such a scene by scene correlation matrix is symmetrical. And this is because the correlation between scene 5 and scene 2 is identical to the correlation between scene 2 and scene 5. In other words, everything I see in the past part of the matrix will be identical in the future part of the matrix. So the way to control for this possible bias of scene similarities, what I'm going to do is actually subtract the upper triangular part of the matrix from the lower triangular part. Everything that is similar, that is related to scene similarities, will be lost. And I will be left with this asymmetrical component that represents reactivation seen at event boundaries with previous or future scenes. So for example, if this is again the real data from the brain, I've subtracted the future part from the past part, and you can see that in this matrix, I have a lot of warm colors, and that means that overall I see more events that are reactivated from the past part of the matrix compared to the future. So this here is a contrast. I'm subtracting the average correlation of the future from the past. If I have more correlations in this part of the matrix, you will see yellow colors. If I have more correlations in this part of the matrix, you will see blue colors. And my hypothesis is that I will find reactivation of past events. Therefore, we would expect to find yellow colors. So this is just the first step. I want to know whether I would see reactivation of past events in the brain. So this is just averaging across all these entries of the matrix. So I remind you that this analysis started with a single voxel and a sphere around it, which created this matrix. So for this voxel that started the analysis, I'm going to assign this difference between the correlation coefficients, and I'm going to give it a color to represent the value, this value of the difference. And then I'm going to go to the next voxel and do the same analysis, create my matrix, do this calculation, and the next voxel, and the next voxel, and so on. And this will give me a map. This map will show me brain areas that show more reactivation of past events compared to future events. Which brain areas would they be? So this is the first data set. It's the movie data set. And you can see a lot of brain areas lighting up. But specifically, you can see precuneus here. You can see angular gyrus. And you can see hippocampus. So that's the first data set. The second data set is the story data set. And here you don't have a lot of sensory areas, as you would expect, by the way. For example, here you have no visual input, right? In the movie data set you do, in the story data set you don't. But here what you see consistently is precuneus, angular gyrus, and hippocampus. So although there are other areas in the movie dataset, I'm not going to interpret them. I'm only going to interpret areas that are replicable across my two datasets. And I can also pull these two datasets together because I can. And that obviously gives me very strong effects in precuneus, hippocampus, and angular gyrus here. Okay. So it seems that default mode regions show reactivation of temporally remote past scenes at event boundaries. And I remind you that these two datasets are so different that we were actually astonished to see this replication across these two datasets. So this is really a very strong effect. Okay, so we have reactivations of past events in default mode areas. And the next question we wanted to ask is, are event boundary representations shared across brains? What do I mean by that? So far I showed you an analysis within a certain brain of a certain participant. So I would take this participant, I would extract scene representations and event boundary representations, I will create my matrix, I will subtract the future from the past, and I will create my map. But previous work has actually shown that while you are all listening to my talk, then the representations in your brains of the events of my talk, like the slides of my talk, are actually shared. There is a correlation between the representations of events between all of your brains. So we wanted to know if not only scene or event representations are shared, we wanted to know whether event boundary representations are also shared across brains. And why is this cool? I'll explain. So first, what are we doing? Now I have the same timeline, but I'm going to take my scene representation from one participant, and I'm going to take the event boundary representations from all other participants in the same data set, and I'm going to average that event boundary representation. So I'm going to do that to create my matrix that now scenes come from one participant and event boundary representations are averaged across the remaining participants. And I'm doing that for all participants. And why is this interesting? Think about my hypothesis for a minute. I'm proposing that at event boundaries, you're uploading previous relevant representations from memory to help you understand what I'm saying. So an event boundary representation can be thought of as this summation of previous representations into a new representation that represents your understanding. So if I'm running this analysis and I'm showing you that event boundary representations are actually shared, and I can find reactivations compared to scene representations from single participants, that would mean that there is also shared binding. The fact that we have shared event boundary representations means that we are choosing the same or very similar events to bind together during event boundaries, and that would support a mutual understanding of a narrative. In other words, if I'm doing a good job, you're all understanding what I'm saying right now because your brain is choosing the right bits of information to reactivate from memory. So will I find these shared event boundary representations in my reactivation analysis? So I'm going to take this matrix created between participants. I'm going to compute the past minus future. I'm going to assign it to each voxel across the brain. And what do I find? So these are the previous results from the within participant analysis. And these are the between participant analysis. And they're very, very similar. So again, in the Sherlock dataset, that's the movie dataset, we have precuneus, angular gyrus, and hippocampus. In the story dataset, again, precuneus, angular gyrus, hippocampus, and in the pooling of all participants, I get the same event. So it seems that event boundary representations are indeed shared across brains, suggesting that we bind information in a very similar way. But what's special about event boundaries? So I'm standing here telling you about event boundaries, and all my hypotheses are about event boundaries, but maybe there is nothing special about them. Maybe any other time point in the movie would show me the same reactivation. So is this reactivation specific to event boundaries? And to answer this, what I'm going to do, so this is my timeline again, you know it, this is my matrix, past and future, and this matrix is based on event boundaries. I remind you, an event boundary is a single point in time, the transition between scenes, and it's the correlation between event boundaries and scene representations. But I can create the same matrix for any other time point in the movie. I can create it for the previous TR, before the event boundary, right? And here I will have the correlations between event boundary -1 representations compared to scene representations. And I can do that for 2 TRs before the event boundaries, creating another matrix, and 3 TRs, and so on. And I can do that for future event TRs after the event boundaries, for event boundary +1 or +2 or +3, and so on. So now I'm creating all these matrices showing me the amount of reactivation I have at different time points in the scene. So so obviously I can compute this one reactivation index for each matrix, average past minus average future. And I'm going to be plotting that for the 3 areas that showed me significant and replicable effects. precuneus, angular gyrus, and hippocampus. So I will be presenting on the x-axis, the leg from the event boundary. So 0 is an event boundary. And on the y-axis, you're going to see the level of reactivation I find as determined by this index, past minus future. Now, I'm reminding you how the bold signal looks like. It starts with a stimulus, and then the bold signal rises and peaks at around 6 seconds after the presentation of the stimulus. Now, this analysis is, I'm not going to show you the bold signal. I'm going to show you the reactivation signal, but it's based on bold measurements, right? So I'm expecting that after an event boundary occurs, an event boundary is just like a stimulus, then its neural signature would occur exactly 6 seconds or 40 hours afterwards. So I'm expecting to find a peak of reactivation just around here if there is indeed something special about event boundaries. Okay, so what do I find? This is the movie data set. You can see precuneus, hippocampus, and angular gyrus. In red, you see this event boundary. It is not corrected for the HRF. And you can see the leg from event boundaries. On the y-axis, you have the level of reactivation. It's not a bold signal. It's the reactivation signal. And you can see that the reactivation peaks just around this 40 hours, which is 6 seconds. This is highly significant for precuneus. It is not significant for hippocampus, and it is significant for angular gyrus. Now, the second data set, the story data set, again, in precuneus, hippocampus, and angular gyrus, we have this peak of reactivation precisely 6 seconds after the onset of the event boundary. I can do that within participants. I can do that between participants. Again, all results are significant. In all 3 areas, I find that there is indeed something very, very special about event boundaries. This is where we have this peak of reactivation, which decays on both parts, on both sides of the event boundaries. Another important thing to say about this analysis is that throughout this talk, I've been showing you different maps, and I'm telling you, well, you see, this is precuneus in one data set, and this is precuneus in another data set, and you have to believe me that these are the same areas. In this analysis, I'm actually cross-validating my results. So because I have 2 data sets, I'm using the story data set to define my regions, and I'm testing them on my movie data set. And then I'm doing the opposite. I'm taking my regions from the movie data set, testing them on my story data set. So overall, I have a cross-validation, showing you that the same areas that show these reactivations that are specific to event boundaries are consistent across the 2 data sets. Cool. So last question that we asked, is this reactivation content specific? So when we're at slide 4, and I'm telling you what's still missing and how I propose to solve the problem, you should definitely reactivate what is known. You should definitely reactivate the intuition to the big problem. You shouldn't be reactivating the joke. The joke is irrelevant. It shouldn't be part of your model, of your understanding of my talk. So is this reactivation that I'm showing you, does it choose the right events to reactivate to support your understanding? How do we go about this? Well, we're doing a textual analysis. We have this text showing us, actually describing all the dialogues in the movie and actually everything that goes on the screen. So for each TR, I know exactly what's going on the screen. I know what is being said and what people are listening to. So this is for the movie data set. And for the story data set, it's actually very simple. I have the text of the narrative. And I'm taking these texts and I'm entering them into a language model. And why am I doing that? I want to create a model that tells me which events are semantically related to each other, which events should be reactivated to support our understanding. So under the assumption that events that are related to each other would share words like the names of characters, like locations, like motives, then I should be able to use language models to detect these links between events. So this is the model created for the movie dataset. You can see that some scenes are more related to each other compared to other scenes. And the big question is this model that tells me which links are created or which scenes are related to each other, would these scenes also be reactivated in the brain? So at a certain event boundaries, would I see the relevant scenes being reactivated more than irrelevant scenes? So what I'm going to do, I'm going to correlate the lower triangular part of my content similarity matrix with the lower triangular part of my brain reactivation matrix. I'm going to do the same for the future part, and I'm going to subtract the two again to avoid this artifact, this possible artifact of scene similarities. So I'm asking, is brain reactivation modulated by the semantic relation between scenes? Are scenes that are relevant being reactivated more than scenes that are irrelevant? I'm doing this past minus future, assigning that to my voxel, and I'm creating a map. And this time my map will show me areas that show a selective reactivation of information. So what do I find? I have a within participant analysis, I have a between participant analysis. And for the within participant analysis, for the movie dataset, I get basically the same regions as I showed you before, including precuneus and angular gyrus and hippocampus, which you can see, and some sensory areas too. For the story dataset, which is more problematic, I get very weak effects that don't survive correction for multiple comparisons. There is some trend in the precuneus, but it's not significant. For the between participant analysis, I get significant results across datasets in precuneus and other areas in the movie datasets and in precuneus for the story dataset. So it seems there is something maybe about this precuneus that selects the right information to reactivate. But the key question is the same precuneus that reactivated events on average, as I showed you before, is that the same area that also reactivates selective events, the right events that are semantically relevant? So the contours that you see here on these maps are the precuneus definitions from the previous independent analysis. And I'm going to run this ROI analysis, showing you that across data sets, across analysis methods, the precuneus actually selects the right events to reactivate in the brain while people are either watching a movie or listening to a story. Okay, so final event boundary. So I showed you that reactivation of remote past events in regions of the default mode network. I've shown you that both within brains, while doing my analysis within a single brain, and also between brains. And the fact that I showed you this effect between brains suggests that there is shared binding of past with present information across people. I've shown you that reactivation is specific to event boundaries, as I showed you in this reactivation time course. And I showed you that reactivation in the precuneus is specific for relevant semantic content, as I demonstrated in my NLP analysis. This means that there is relevant binding of past with present information. Now, these are very robust findings that are replicated across two very different data sets and two different analysis methods. And this is, I think, interesting because reactivations were suggested to have this offline function. We chunk events and we just store them into memory. But here I'm showing you that they operate on the fly while you are experiencing a movie or a story. And they're also processed by this presumably offline regions of the brain. So I'm actually proposing that perhaps we could think about replay and the default mode network in a different way. I think that reactivation may be an online mechanism that binds information across time to interpret the present experience in light of past events. And it operates on the fly. And once it does that, yes, it can consolidate event into memories as a byproduct. But as a first function, it helps you understand what is happening in your daily experience. So this I think is very informative, both to human and rodent literature. Cool. So if I did a good job and you had all the right replays at event boundaries, then I hope that this narrative made sense. I want to thank Tim, my supervisor, Chaim, who collaborated with me on this project, and Tasha, who did this, who's doing this ongoing work. I want to thank my funders, the Marie Curie, Human Frontiers, and EMBO agencies, and thank you for listening.
Avital, thank you very much. This was really, really interesting. I'm asking now everybody, you can unmute yourself and let's give Avital a big applaud. Really interesting. And there was one, so Kaula Al-Kadi said that I have a question. Thank you. So do you want to, if Avital is happy with that, maybe you want to unmute yourself and
Sharon,
ask?
should I unshare? Should I stop sharing?
Yeah, you could.
Okay.
Hello, teacher. Hello, doctor. Thank you for your presentation. It's really incredible. So I am Khawla, PhD student in neuroscience, but specifically I work about Parkinson's disease. So what do you think about the impact of some therapy? For example, cognitive behavior therapy for to active some area at the brain. For example, a long term and or short term. And thank you.
Sorry, could you repeat the question? I couldn't quite hear that.
What do you think about the impact of some therapy? For example, CBT, cognitive behavior therapy for to active some area at the brain.
I see. So, so if I understand correctly, you're asking me about could this be applicable for therapy or for different disorders? Is this your question?
Yes, doctor.
Okay. So I think it's a really interesting question. I think there are many disorders that are related to this aberrant reactivation, or not reactivation, but the aberrant ability to bind events into time. The most obvious example is Alzheimer's, right? So when people with Alzheimer reach the hospital, they often can't understand how they got there because they can't bind the previous events that led them there. So it is definitely possible that they have this problem in the reactivation of relevant information on the fly, and that hinders their ability to understand the current experience. And there are many other clinical disorders that could be related to these kind of reactivations. You can think about ruminations, for example, in anxiety disorders, where people constantly, they're stuck in this loop of thinking, what's going to happen? What did I do wrong? And what, like, this constant loop could be related to the constant reactivation of information that can't stop. So as for your question about therapy, if indeed this is true, then if we could somehow silence this aberrant reactivation in ruminations, or somehow enhance this reactivation in Alzheimer's or dementia, then maybe that could help people. But this is, of course, merely speculation.
Thank you, doctor, for your answer. And if you are interested about the subject, I will send you an article about this subject.
Thank you.
You can just write your email, your address email, if you are
Sure.
if you don't have any problem.
Sure.
Paula, you can write to me. I organize the seminar and I will pass that to Avital, okay?
Okay. Thank you.
Okay. Avital, are you up for more questions?
Of course.
I see that Hagit, I also have my own, but Hagit, yeah, you want to add me just, yeah.
Sure. Okay, so thank you, Avital. A really amazing talk. I really enjoyed it. I'm from the computer science department, and I see a lot of similarity in the things that you found in the brain to what we call transformers. I don't know if you, right? It's a just for the others, it's a deep neural network where one of the elements is a matrix that tries to combine or relate different parts of the sentence or a sequence.
Are you referring to the attention matrix?
Yes.
Okay.
Yeah. Yeah, I just want to raise the question if you've thought about the similarities there.
I am
think it's
constantly thinking about these similarities, and I'm really glad that you raised it. I'd be happy to discuss it with you too. So I'm not an NLP expert at all. But to the best of my understanding, current NLP models don't understand. They're very good at memorizing, but they don't quite understand. And the way I define understanding is actually very similar to what I presented here, is creating the right links between events that form this internal model of understanding. So I'm not sure that attentional weights actually, it's an open question, right? I don't know if these attention weights actually form these right links between events. I kind of suspect they don't. They also operate at the level of words, right? At the level of tokens rather than at the level of GIFs. So I think for a language model to actually understand, it would need to do several things. It would need to first know how to parcel an ongoing experience into events. It would need to abstract these events into gists, and it would need to link these different gists in a meaningful way to construct this model that I will call understanding. I'm thinking about these ideas with people like Chaim Dubosarski from the Queen Mary University and Maria Leakata from the Allen Institute. I would be really happy to hear your thoughts.
Sure. It's always a question, I mean, sort of a philosophical question, whether the GPTs are actually understanding. So what is understanding? Do they just the fact that they know that they come together, or they are some sort of associated in a sequence, or in many of the sequences, does that mean understanding or not? I mean, it's sort of a philosophical question, I guess.
Yeah, so my collaborators tell me they don't actually believe that the ChatGPT understand. It's more, it memorizes. And the longer the text becomes, it loses context. It just can't keep track. So this is more, I think, of a memory.
Yeah, nowadays, now it's doing much better in that, for sure.
Okay.
But again, it's a question of what do you call understanding? Just
Yeah.
the fact that it's related somehow, is that understanding, or do you need a sort of higher level of connectivity between them? And I'm wondering, I mean, maybe the whole GPT kind of LLM approach needs to go up a level and look at, as you say, chunks versus
So I think there
or
are
tokenize it different. Maybe we have to tokenize it differently.
Yeah. So I think there are NLP applications that now are able to chunk texts into events, and they represent them in a meaningful way. I haven't seen any links created between these chunks of text. I think that will be a big step forward.
Yeah, well, the dimensionality goes up like crazy.
Yeah.
Hey, thank you. It was a very good talk. I enjoyed
Thank
it
you
very
very
much.
much. Thank you.
I myself have a question about, so, there's the binding problem of
Yeah.
where do we bind the information together, because we talk about, separating the information into different channels, and then it just binds. And I'm wondering if, I don't know if it's these event boundaries where we bind it, or do we have more event boundaries that are not, only at, scenes, or do we always, I mean, do we create them? Or maybe, Ayelet Landau showed that attention, fluctuates. Are these attentional fluctuations, in fact, related to creating like artificial event boundaries to allow us to, to bind things, because if I see a green shoe, I'm not gonna wait until, to understand that it's green. I understand that it's green when I see it and that it's a shoe. So maybe these fluctuations allow us to, I'm just thinking.
Yeah, I completely agree. Actually, so my definition of event boundaries was related to the areas that I wanted to study, which are the default mode network. And we know that these areas have a large temporal window. They process information across big chunks of time.
Yeah.
Now, you could be asking, well, what would be an event boundary for visual cortex? For visual cortex, it doesn't make sense, as you say, to wait until the end of an episodic event to define what a red shoe is. So there is actually work by
Green.
Chris.
No, I'm joking. Yeah. No, I said green.
Sorry. Attentional left.
Chunking. Yeah, okay.
There is work by Chris Baldessano who used the movie data set and showed that different brain areas have a different length of temporal windows. So when you move from primary areas to high order areas, the window size increases. So V1, for example, would have a very short temporal window because the stimuli, the input it cares about, comes in short time scales. So it would mark an event at the changes in context that are specific to V1. So that would be, for example, different orientation. But as you move up the hierarchy, different areas would define changes in context in a different way, and that temporal window would increase. So I think, and this is again hand waving, I think that these event boundaries we would see these event boundaries at different levels of integration, but they will come at different times. And that would depend on the kind of input that a certain brain area expects to process.
Interesting. So really, Uri Hasson had a paper about, different temporal time windows. I mean, maybe it's the same paper that you are referring to, but
Yep.
so it could be really interesting if this is linked. And I also wanted to ask, you did the correlation between participants. And I wanted to ask because, we can both experience an event and I can remember something very differently from what you experience.
Mhm.
And the fact, so I'm wondering if there's some sort of analysis that examines how, for example, I correlate the information of past. I mean, what I'm saying is that if you look between participants, maybe that also adds noise because we do have different memories or
I agree with you completely. I think mostly in the literature, we're looking at shared representations just because that gives us a better SNR. But there's definitely an idiosyncratic element to our experience. I actually developed a method to detect these idiosyncrasies in brain representations. I haven't done anything with it, but it's out there. If you have any good ideas about how to use it, you're welcome to it. But yes, I have found that we have both shared and idiosyncratic representations that operate simultaneously. And their relation to experience, I mean, I'm sure there is. There are some parts that we understand in a similar way, and some parts we understand in different ways. And it's very interesting to try to understand how these two things balance.
wow, this is really fascinating. Just another suggestion last. The work by your student, if she can have a look at the functional connectivity of those voxels in the precuneus to see what other regions lit up. I don't know, maybe you're going to find the hippocampus as well.
I'm sure I will. So specifically for function connectivity, I'm sure I will.
Okay, so Avital, it was really, really great, a great talk and pleasure having you. And again, thank you so much for joining us.
Thank you. It has been a pleasure. Thank you.
Thank you very much.
Very nice.
Thank you, Amos.
Thank you. Oh, and I want to say, I'll let you read the chat. Sarit says she has a question. Sorry,
Okay.
Sarit, I haven't, so if you were, is she here? No, I missed that. Okay, so I get, she said great talk, but I'll connect the two of you. So she can ask you later.
Cool.
Sorry.
Thank you, Siobhan, and thanks everybody.
Thank you. Bye-bye.
Bye-bye.